Good morning, everyone. Thanks so much for coming. I'm really happy for this opportunity to share my master's research with you. The title of my presentation, as well as my thesis, is Enhancing Native Plant Diversity on Legacy Minelands. And before diving into the details of my research project, I want to set the stage for the details of my research. I want to set the stage by sharing some background info on my research site. Before coming to the University of Kentucky, I spent 4 years working in West Virginia for the U.S. Forest Service on the Monongahela National Forest. And my favorite project was our large-scale ecological restoration project on Minelands. It's a collaborative project with many partners involved, and we've been restoring and reforesting from 100 to 250 acres each year since about 2007. There are 2 sites on the National Forest that are doing this ecological restoration work on Minelands, and I did part of my research on both, but I'm going to start with the site on the district where I worked, and it's called the Mower Tract. The majority of my research fieldwork took place here on the Mower Tract, and it's located in eastern West Virginia in the heart of the Monongahela National Forest, here in green on this map. And it's 40,000 acres that were purchased by the National Forest after a series of disturbances. So in pink on this map, these are the 6,200 acres that were mined. And then in green, these are the areas that have been restored so far since the beginning of the project. And the reason that restoring these minelands is so important is because they're part of the red spruce ecosystem that used to dominate the high elevations of Appalachia. Red spruce forests of Appalachia are biodiversity hotspots, and in West Virginia, they support 105 state rare plant species and 137 wildlife species of concern. The problem is that the red spruce hardwood forests once occupied 500,000 acres in West Virginia, but a series of disturbances, including extensive logging of the area for the pulp and paper industry, and then fires that were sparked by trains moving lumber in and out of the area, they burned the thick layer of duff and spruce needles and leaf litter and led to the loss of the seed banks, especially of the red spruce seeds, and then also caused extensive soil erosion. And only about 40,000 acres of the original red spruce community remain. The other problem, which caused the loss of the red spruce forests, was the extensive surface mining for coal. On the Mower Tract, 2,600 acres were mined in the late 1970s, and in Appalachia, 1.5 million acres have been surface mined. And the mines, we call them legacy mines, the mines that were reclaimed after the 1977 mining law, and they were reclaimed to a state that is not similar to their pre-mining condition. So to give you an idea of the damage that surface mining causes, and to show you what we have to work with on these sites, take a look at these photos on the left here. These are both on the Mower Tract, circa about 1980 during mining. And then on the right here, that is what's right at the surface underneath. This is after decompaction. It's these mine spoils, which is the blasted rock layer from above and below the mine seam. And it's just a mixture of rock, and they end up mixing it and ending up burying the topsoil. So this is what we have to work with to plant our trees in. So then they heavily compact it and plant it with plantations or grasslands. So the post-mining landscapes look like this. They're originally reclaimed to either non-native conifer plantations or grasslands that really look like pasture. Both of these photos are taken 40 years after the original reclamation. And as you can see in this plantation picture, these trees are really, really small for 40 years old. They're highly stunted because of the compaction that this land and then in the understory, as you can see, there's little, if any, understory diversity and really no mid-story at all. And then in the grassland pasture, these trees were planted by originally by the mining company, but there are no other trees growing, as you can see. They're stuck in, both of these lands are stuck in a state of arrested succession, which is the ability of disturbed land to develop through natural processes and the resulting failure of later successional species to colonize the site. So these lands are damaged, and without intervention in the form of restoration, they may never function as natural ecosystems. So the plantations are monoculture, just one type of tree, and then in these grasslands, they used to seed just maybe 5 or 6 different species of grasses that are really aggressive they grow quickly and then they prevent erosion. But really the problems on these mine lands go beyond the low plant diversity. There are very low soil nutrients because of the mine spoils. As you see, they're mostly rock, and very low organic matter, and then poor water infiltration because of the compaction. And also these aggressive grasses end up competing with native species, and that's why, as you can see, it hasn't developed through natural succession and become a forest again. So the overall goal of the ecological restoration work on the Monongahela National Forest is to restore a functioning ecosystem that provides at least some of the ecosystem services that it did before mining. So we're reforesting to bring back the red spruce forests, and this is going to help bring organic matter back into the ecosystem and help the land to start producing soils that cycle nutrients again. And of course, reforestation mitigates some of the effects of climate change. And with the decompaction work that we do, we're improving the watershed and allowing the water to start infiltrating down into the soil. And with the native plants that we're planting, we're providing habitat to support local plants and wildlife. So I'll tell you about the process that we go through for this mineland restoration. Site preparation activities are the activities that prepare the land to be planted with trees and shrubs. And the process is a little bit different on the lands that are grasslands, like the picture on the left, versus the minelands that are non-native plantations on the right. So for the grasslands, they bring in 2 D8 or D9 dozers, and they go through and they're decompacting, they're ripping up or tilling the land with these dozers. On the back of these dozers, as you can see in this picture here, they pull a 3-foot ripping shank. And on the sites with plantations, they push all the non-native trees down, and then they go through and do the ripping. And then we bring in an excavator, and because the whole hydrology of the mountain changes after we do this decompaction and then ripping, water begins to settle in different areas, and we bring an excavator and it digs, it creates wetlands and vernal pools all over the site. And then also he spreads back the trees, he spreads them out all over the whole site. And I'll talk more about this process and why it's important in a little bit. And then after this process, the following spring, we plant trees and shrubs of as many species as we can get our hands on. To give you an idea of some of the numbers, in 2020, they planted more than 98,000 trees and shrubs on 200 acres just on the Mower Tract. And they so 320 pounds of native seed were sown, but then totals to date, they've restored so far 964 acres with more than 452,000 trees and shrubs, and they've created more than 1,500 wetlands. And then normally each year after we have this restoration, we bring in volunteers and we through green forest work, and we've brought in more than 500 volunteers just in the Mower Tract alone. So the restoration work so far has been successful, and we've been seeing that after decompaction, native plants are able to colonize the site, and planted trees are able to grow and survive a lot better. Take a look at this top photo. This was taken just last week in West Virginia on one of our earliest sites called Barton Bench. And these trees, I believe they're 7 to 10 years old, and they're doing really, really well. And then this photo down here, this site was ripped in 2014 and planted in 2015. I believe it's Lambert South. This was a site with all the woody debris, so you can see a little bit of the woody debris. And then we planted a lot of aspen, but then a lot of the trees that are coming up in this young forest now, our fire cherry, which we don't plant, and there are a lot of other species that we don't plant because the wildlife is coming in and utilizing the site and helping to spread seeds. So it's looking really good, and it's already becoming a young forest. So, but we have confidence in our reforestation efforts, but we've had more questions and more conversations. Are we taking full advantage of the early successional habitat potential? And then we've seen a couple of different problem areas, for example, sites that have these a little bit barren slopes, and we've wondered what the best way to deal with these are. So we think that there are more opportunities before the trees grow up into the canopy. We think there's more opportunities for early successional habitat and soil building, and especially for increasing the native plant diversity. So our early efforts to increase plant diversity began with our partnership with the Natural Resource Conservation Service Plant Material Center in Alderson, West Virginia. In 2007, the Native Plant Program began as a native species propagation effort to supplement the diversity of species that we plant on the Minelands. Every summer and fall, we collect seed locally from high elevation red spruce forests all over the Monongahela National Forest, so both uplands and wetland areas like this bog here. And we work together with our partners at the Plant Material Center to clean the seed, germinate it, and then pot everything up. And then once they're ready, like you can see down here, we end up planting these back on the Minelands. So through this program, we've been able to increase plant diversity, but the Plant Material Center is a very small operation. So the extra seed that we had, we would end up spreading on the newest stripped areas. And anecdotally, we've seen a lot of success from seeding, but we never tried it on a large scale because seed is very expensive, and we've never really tested this scientifically to find out which species would even be able to do well on these Minelands. So this is where my research comes in. I love this Mineland restoration project so much that I came back to school to learn ways to enhance our ecological impact. We plant tens of thousands of trees and shrubs each year, but my coworkers and I always thought that the understory, the herbaceous layer, was a missed opportunity for wildlife and pollinator habitat in the years before the trees grow up into the canopy. So for my research, I wanted to focus on herbaceous plants, especially those that benefit pollinators. I wanted to learn how to increase the diversity of native herbaceous plants on a large scale on these Minelands. There are many benefits to increasing native plant diversity on Minelands. The red spruce ecosystem, as I mentioned, is a biodiversity hotspot. So we want to restore the connectivity and bring back these fragmented disturbed lands. We want to reforest them to red spruce forests. And using native plant diversity, native plants are adapted to local conditions, and they've evolved alongside local wildlife and insects. So those are the best ones to use for restoration because they are able to increase the resiliency of the restored ecosystem. And using more plant diversity attracts more diverse wildlife and insects, which end up further restoring the site for us. By bringing seeds and utilizing the habitat, they end up adding more benefits to the restoration work. The benefits of herbaceous plants, especially, provide many benefits in restoration. They produce organic matter, they store organic carbon, and they produce ground cover, which retains moisture. And they offer food and habitat structure for wildlife. The different sizes of plants and different heights provide a lot of structure for habitat. And then flowering species produce pollen and nectar, and many serve as larval host plants for pollinators. So my research bench, my research began with 4 major research questions. And the first one was, which flowering species native to the red spruce ecosystem are viable candidates for direct seeding on legacy Minelands? And to answer this question, I did seed trials. Botanists and ecologists on the Monongahela National Forest helped me select which species to test, and our criteria was they needed to be important to the red spruce ecosystem, they needed to be available commercially, and I needed them, because of the short nature of my master's project, I needed them to germinate in the first growing season. And because of the aggressive grasses on the sites, they needed to be taller than 1 foot. And I wanted them to be flowering species that would be important to pollinators. So these are the species we came up with to use. There are 9 herbaceous species and 1 is a woody species, the sumac. So tall white beard tongue, black-eyed Susan, butterfly milkweed, evening primrose, purple node Joe pieweed, common milkweed, New York ironweed, staghorn sumac, showy tick trefoil, and world rosinweed. So the bench that we chose for my research project on the Mower Tract was decompacted late summer of 2018, and in October we established 72 1 meter by 1 meter plots. So each species was seeded on its own. There were 6 plots for each species. And about twice a month, we monitored these plots, and new seedlings were marked, dead or disappeared seedlings or browsed were tallied, and later all the competing vegetation was identified. So we measured germination, percent cover, and survival. So this data was collected and analyzed. But I'm going to pause before I continue talking about the plots, because these plots were also used to answer my second research question. I wanted to learn, does the addition of woody debris impact seed germination, cover, or survival? And the reason we wanted to learn about this was because on the plantation sites, like I showed you the pictures, each year we had been pushing down these non-natives and spreading them back over the site, and we wanted to know how they impacted. We had seen anecdotally that they're helping our trees to grow better, but we wanted to know if they would have an impact on seeding, and if there would be different species that would be better off seeded in an area with woody debris versus on an area that is barren, just bare soil. So we had all that, we have information anecdotally, but we wanted to test this experimentally. So half of all the plots for each species, so 3 plots for each species, were covered in 5 kilograms of fresh red pine branches and needles, and this was done to mimic the woody debris that we scatter over the mine sites on the ribbed plantations. And these are the plots, as you can see. And there are so many benefits of utilizing woody debris in mineland restoration. Especially on these slopes, it helps to slow down soils that might begin eroding down the slope. When we plant trees in and around the woody debris, it really helps them survive by providing a little microhabitat. So it protects from wind and weather events and hot and cold extremes. And it provides an obstacle to deer that come in and like to browse our trees. And it provides a lot of habitat structure, provides shelter, perches, diversifies the structure, provides hiding spots. It brings in a lot of wildlife, insects, which bring in more birds and other wildlife, which help to spread more seeds on our restoration areas and further perpetuates the restoration for us through the years. And also, as it breaks down slowly, it's providing nutrients to the soil and helping build the soil organic layer. So I'm going to go over some of the results of this part of my research. I did look at each species individually, the differences between no wood plots and wood plots, but for the sake of time, I'm going to show you just grouped results. I found some general trends for germination. NW is the no wood plots and WD is the woody debris plots. For most of the species, grouping everything, all the species were grouped into no wood group and woody debris group for this part. And the no wood plots did better, and I think that's because for germination, the seeds need the warmth of the sunlight to germinate. And then for cover, the woody debris species did better, but there was not a statistical significance. There was also not statistical significance for the germination. But then for survival, the woody debris plots had higher, the 10 species examined in this study the woody debris improved their overall survival, and it was statistically significant. It improved it from 55% with no wood to 74% in the woody debris plots. And I believe this is because of the microhabitat that I mentioned, keeping the temperature low and keeping the moisture higher. So I also looked at results, as I said, I looked at results for species overall, but now I looked at, regardless of woody debris or no wood treatment, I wanted to see how the species did overall. So the letters represent statistical significance. So the A is statistically different from the B, which is statistically different from the C. But anyway, for germination, showy tick trefoil did best, and that's a nitrogen fixer. So it has an advantage of producing its own nitrogen, and that's advantageous in sites that are disturbed and have low nutrients. And for cover, we had across the board pretty low cover for most of the species, and that's not surprising because it's just the first growing season, but time will tell in the future how these species do as far as cover, but they were really, really tiny even at the end of the season, so that's okay for the first growing season. But BE, which is black-eyed Susan, did best, and then EP, which is evening primrose, followed by NY, which is New York ironweed, did best with cover. And then survival, overall, there was really high survival across the board, which was really promising. And it shows that the species that we tested all have varying degrees of potential for use on Minelands. But I wanted to note that the survival was calculated just by using the seedlings that germinated, because I followed them through the whole growing season. So this is a little tricky. Because we had 10 species, 2 treatment types for the plots, and then we measured 3 different things, we had 60 different results, and we needed to come up with a way to make a general recommendation of which species do best in areas with woody debris and which species do best in areas with no woody debris. So we came up with this ranking system, just ranking from 1 to 10, and then we put everything together and we came up with the best species for the no wood treatment. For no wood seed mixes to be used in areas with no wood would be evening primrose, tall white beard tongue, black-eyed Susan, and butterfly milkweed. And then the best species to use in areas with woody debris were New York ironweed, world rosinweed, evening primrose, and black-eyed Susan. And there were 2 species that showed up in both, evening primrose and black-eyed Susan, so they're pretty good generalists. They could be used all over the site. But if land managers did not want to have separate seed mixes, we also came up with combined rankings. So no matter of woody debris or not, you can use these species, and the top ones were evening primrose, WR is world rosinweed, BE is black-eyed Susan, and New York ironweed. And the species that did not do best, that were ranked the lowest in the no wood treatment, Joe pieweed did the poorest, followed by milkweed. And then in the woody debris group, tall white beard tongue, followed by Joe pieweed, performed the poorest. And then Joe pieweed also performed the poorest in the combined group, followed by milkweed. So my third research question was about the seed bank on these native sites, because we see a lot of changes after decompacting. A lot of species are able to sprout up, and we wondered what if native plant species are trapped in the seed bank on these legacy mine sites. So like I started to say, when we go in and decompact these lands, this is a before and after picture of a site on Sharp Knob, which I'll talk about later. It's the other Mineland restoration site. We see a lot of diversity popping up after we decompact these sites, and we'd been noticing this phenomenon, but we didn't have any data to back it up. So we were wondering if these seeds have been sitting in the soil seed bank or if they're being dispersed to the site after the ripping. And we think that while these lands are stuck in arrested succession, like I said, they're compacted and covered with these aggressive grasses, that this might be preventing them from growing. But then whenever we decompact and break that arrested succession, we're thinking that that might be why. So that's why I wanted to study these questions. And also on the mine bench that I used for my research, it's very narrow, and above it, there's adjacent native forest above it that I thought was providing a good seed source. So most of our minelands have some native forest adjacent. So I wanted to see what species were seeding in from the native forest also. So to answer this question, I did a seed bank characterization, and I did vegetation assessments and botany surveys in different types of minelands and also the adjacent native forest. So we took soil samples before my research bench was decompacted, and I brought the soil back to Kentucky and separated out the moss and plant material from the soil, and then cold stratified these in a refrigerator for 4 months. And then soil and moss were put separately into 7 trays each in a greenhouse, and they were kept moist in the greenhouse for 125 days for the spring germination. And they were dried out over the summer, which acted as a ripening period while I was away doing field work in West Virginia. And then the trays were kept moist again for 102 days for fall germination. So 1 to 2 times each week, I went to the greenhouse and went through each tray. And this is an example of what I was working with each week for each tray. I went through identifying, pulling out, tallying, and potting up the seedlings that I couldn't recognize, that I couldn't identify right away if they were too small. This process was really involved and took a lot of time, and I had friends and colleagues help me out in the greenhouse and help me out a lot with the identification of the species, which was really tough. But overall, in the end, after both rounds, a total of 2,022 seedlings grew in the greenhouse seed bank study. And the seedlings were identified to be 55 different species, representing 19 families total. So throughout the process, I was taking pictures to help me learn how to identify these from when they're very, very tiny. And later, I want to try to put together some type of a seedling reference guide, because it would have helped me a lot while I was doing this process. But I'm going to show you 2 examples. One is lance-leaved loosestrife. So I tracked it from when it was very small through its development up until it flowered, when I was able to positively identify it. And then another one was Lobelia inflata, Indian tobacco, from when they're very, very tiny and hard to figure out what they are. And I made all kinds of guesses, but until it matures and makes flowers, it's really, really hard to positively identify. So these are some of the other things that sprouted up in the seed bank study in the greenhouse. We had deer tongue grass. There were several birch trees, which may be yellow or black birch. This was purple sneezeweed. There was a few types of orchid. This one is ladies' tresses, so that was surprising. And then we had some black-eyed Susan, hundreds of different types, or I looked like different types, but they weren't. They were mostly hundreds of types of asters, but most of them ended up being goldenrod. And then different types of hypericum, St. John's wort. This was Lactuca biennis. And then there were a few striped maple trees, and then some weedy ones like the stellaria and all-site clover, which was originally seeded by the mining company. So after the greenhouse work was done, we wanted to learn through talking with Dr. Baskin. She knows that some species might take up to 25 years to actually germinate, and they might be sitting in the seed bank waiting that whole time. So I wanted to find out if there were viable non-germinated seeds that were still in these soil samples. So I took soil subsamples, and Andrea helped me out immensely by going through these and looking for, cleaning the samples and looking for anything that looked like it could be seed. And then I went through and separated them out into different types. It looked to me like there were 19 different types. And then I wanted to find out if they were alive, so I put them in water to imbibe, and then and sliced them in half with a scalpel. And as you can see in this picture, was looking for the white live embryo inside. And then some of them I wasn't sure, so I put them in a 1% tetrazolium mix. And as you can see, this is like a little bit, it's hard to see, but if they were alive and respiring, they turned a little bit pink, like you can see here. So after that, I ended up finding 7 different types of seeds that were alive and had not germinated. But for a later study, I'm going to identify those and find out what species they were, and if they were additional species or if they were ones that had already sprouted in the green bank seed bank. Sorry, not green bank, greenhouse seed bank study. So I thought it would be really interesting to compare the seed bank to what was growing on these mine sites. So my coworkers in West Virginia helped me do some botany surveys. This is a tiny version of the Mower Tract Mineland map, and in red here is highlighted just this map here of my project area. This yellow that's my research bench, the yellow one, and the green is the native forest above my research bench, and then over here in the purple, that is a non-ripped bench. And then the purple lines are our tracks when we did the botany surveys. And we did these surveys, we use the process that the Forest Service uses for botany clearance surveys to do management on new areas. So we just got as much coverage of the area as we could, just zigzagging around, and any new species we would find, we would identify them and mark them down as present. So this was just a presence-absence type of survey. So results of these surveys: on the non-ripped bench, we found 45 species. And then on the ripped bench, we found 108 species. And this was a combination of what we saw and then what I had identified in my plots also. And in the greenhouse, as I mentioned, 55 species came up. And then in the adjacent forest, there were 46 species. And I know that we can't directly compare the adjacent forest to these mine lands, but I was really curious and wanted to see, to be able to compare some of the species. So to give you a better perspective here, the ripped site had 108 species. This is an increase of 63 species from the non-ripped site. And to show you, I was interested in non-native versus native species. So don't pay too much attention to these other ones. These are just different types of non-native species, but then the yellow one is the native species. And I'm really excited about the number of natives that came up in on the ripped mine benches compared to the unripped. And also for the future, I wanted to mention, although the ripping increases the number of species, there were 11 exotic or invasive species that were found before and after the ripping, and there were 4 new exotic or invasive species that came up after the ripping. So we're going to have to keep an eye on these species as far as management of these sites. And for the future, it's of great interest to us to figure out how we can get rid of these non-native species that are seeded by the mining company in the original reclamation mix, because they end up dominating these sites. So I wanted to compare all of these sites, and I looked at Jacquard's Similarity. And what's interesting here is the non-ripped, the most similar sites were the non-ripped site and the ripped site. They had 30.8% similarity. And then the lowest similarity was between the greenhouse seed bank and the native forest, had just 2% similarity. So that was surprising to me. I thought more species were seeding in, but maybe they hadn't germinated yet. So I'll find out with those 7 species. And then floristic quality is determined based on degrees of similarity to the plant community composition of a natural area of high quality. So using the native forest as a reference, the species that I found in the native forest gave it a ranking of 38.2 for the floristic quality index. And then the non-ripped site had 17.4. And then once we ripped it, it was 30.8. So that's kind of exciting too. And this is just in the first growing season. So time might tell whether this stabilizes to a lower number of natives later. We'll have to find out in future growing seasons. So through this study, I wanted to gain an understanding of whether species in the adjacent forest were dispersing seeds onto my legacy mine benches, and if decompaction influenced the native plant diversity. So some of the takeaways of my results are that decompaction increased native species richness by 63 species, which was really exciting. There were 10 native species that were found in the soil seed bank, were not growing on the non-ripped site, but then they did grow on the ripped site. So this answered my question about our native species trapped in the seed bank. And then there were 19 native species that were growing in the adjacent forest that ended up growing on the ripped site. So the seed bank doesn't tell everything. I'm glad I did these surveys and learned that information. And as I mentioned, future monitoring will reveal if this phenomenon is just like a temporary spike in richness in the first growing season, or if it can be sustained into the future. I had hypothesized that native plant seeds were present in the soil seed bank, but not growing on the legacy mines because of the arrested succession. But once we broke that, they were able to grow, so this was confirmed. Seeds are being wind and animal dispersed to the sites also. They're not exclusively from the adjacent native forest. So my final research question, this was kind of a side project that we did. We wanted to learn, will native herbaceous plugs transplanted to created vernal pools on minelands, will they survive and produce cover? So this is the other mineland restoration site on the Marlinton district. It's called Sharp Knob. It's about 20 miles as the crow flies from the Mower Tract. And this is the area that was mined. There was about 1,700 acres that were surface mined in the late 1970s. And in 2018, the site that they decompacted, they constructed 8 wetlands or vernal pools of various sizes. And so we wanted to plant some herbaceous transplants around these wetlands to see if they would survive, because we plant tons and tons of bare root or containerized trees and shrubs, but we never tried with herbaceous plants. So we selected 4 species. It was swamp milkweed, boneset, Joe pieweed, and blue vervain. And we purchased this seed and sent it to our partners at the Natural Resources Conservation Service Plant Materials Center. And they germinated and grew them out for us. And then this is a picture of the plants when they were ready to be planted. And then we had a big work day with Forest Service, Green Forests work, Office of Surface Mining, AmeriCorps, and Greening Youth. And we made 4 plots around each of these 8 wetlands and planted 1 species in each plot. We planted 25 of each species in 1 plot around each of the wetlands. I visited these sites with help about once a month, and we were measuring wetland water height because that fluctuated throughout the season. And then we were going in and hunting for, searching for these individuals planted in these plots, and sometimes it got really difficult. A lot of grasses grew. And by the end of the season, we saw that many of them had flowered, so that was exciting. This one is boneset, and that's blue vervain. So results of this small study, boneset had the highest percent cover. The percent covers were kind of low, but again, it's the first growing season, so that's okay. It might improve in the future. We'll just have to keep monitoring this site. Boneset did best, followed by Joe pieweed. And then as far as survival, across the board, they had really good survival. The boneset had about 80% survival. The blue vervain had about 75. Joe pieweed had about 72. And even swamp milkweed had about 59, which is not terrible for the first growing season, or for transplanted plants in general. Some observations that I made during transplanted, I ended up, especially me, ended up breaking a lot of these herbaceous plants because they're very delicate, and I think that's why a lot of restorationists don't want to use herbaceous transplants, because they break easily. But anecdotally, I noticed that it ended up stimulating their growth, and they ended up surviving anyway. I didn't lose any of these plants because of them breaking during transplanting. These species reproduce through rhizomes, which are horizontal underground stems that produce shoots and roots at different intervals. And this is something common that plants have, and it's a really good quality for species used to revegetate restoration sites that are disturbed, because it allows species to form colonies, which increases cover. So results of my research showed that decompaction not only benefits forest restoration, but it also breaks the arrested succession and increases plant species richness. In addition, from past experiences and some of the literature, we know that it also increases planted tree and shrub survival. Seeding is successful for the species that I tried. It's better for certain species than others, and woody debris has many restoration benefits. And the Mineland plantations, they really have no woody debris on the ground, so it's a missing component of that forest. So by putting it back, we're adding that to these forests and also helping with the ecological restoration of the site. The fact that woody debris is beneficial to the survival of seeded natives, like I found, is just one more benefit of keeping and utilizing woody debris on site on plantations that are restored. And then finally, transplanting herbaceous plants to constructed vernal pools is a viable option for increasing plant diversity in created wetland ecosystems on these restored mines. So future mineland restoration on the Monongahela National Forest. Green Forest's work has identified more than 25,000 acres of high elevation areas that are suitable for red spruce restoration on or adjacent to the Monongahela National Forest. This is the National Forest here. And then across Appalachia, there are still 1.5 million acres of legacy minelands that are in need of restoration. So I'm really hopeful that the results of my research will help guide future restoration efforts on the Monongahela National Forest and on other minelands in Appalachia, especially high elevation minelands. So I'd like to conclude by thanking so many people that helped me with my project. My committee members, Dr. Barton, Dr. Baskin, Dr. Agaritas, and Rob Ratley, the Department of Forestry and Natural Resources, and all the Forest Service support that I had, Shane and Jack, Amy and Cindy. And I had a lot of help with my research proposal and organizing my ideas, lots of help with my seed trials. So many people helped out with my seed bank study working in the greenhouse, and Wendy and Allison were a huge help, especially Allison. She tutored me through my statistics for months. I am indebted to her. Had a lot of help with my botany surveys, and with the vernal pool transplants was a huge effort. So with that, I thank you for listening and being interested in my research, and I'd be happy to take any questions.